Nikki, can we start? Okay. Good early afternoon. Welcome to the 11th Fred Chaoul Annual Workshop Meeting. My name is Natan Shaked. I'm chairing this conference together with Vered Padler-Karavani. I'm coming from the Department of Biomedical Engineering. I'm heading an optical interferometry and nanoscopy laboratory. We have a distinguished program starting from an invited plenary lecturer who Vered will introduce in a second. I'd like to ask all the lecturers in all the sessions to come before the presentation to upload the presentations to the computer. And I would like to congratulate you for attending this program, and I hope you will enjoy it. Vered.
Great, welcome everybody. It's a pleasure to have you all here, and it's a pleasure to welcome Peter Seeberger for the keynote speaker. So I'd like to say a few words about Peter. He studied chemistry in Germany, and then he did his PhD in biochemistry in Boulder in USA. He did some research in the Sloan Kettering Cancer Research Center, and then became an independent research scientist at the MIT, becoming an associate professor. And then he became full professor at the Swiss Federal Institute of Technology in Zurich. And in 2009, he became the director of the Max-Planck-Institute for Colloid and Surface in Potsdam. Peter published over 400 peer-reviewed papers. He has several books, more than 35 patents, and from what I hear, he's going to double that in a short time. More than 700 invited lectures. And of course, he has many honorary awards, but to name a few, from Israel, he got the honorary lifetime member for Israel Chemical Society, as well as from Switzerland, the 100 most important Swiss. In 2013, he was also elected the Berlin Brandenburg Academy of Sciences. And not talking only about science, he also is a co-founder of the Hope for Africa Foundation, which aims to improve health in Ethiopia, healthcare in Ethiopia. And of course, his research also matured into several companies, both in Germany and in the US. So Peter, please, welcome.
Touch it, okay. It's a little naval. So thank you very much for the kind introduction, and I'd like to thank the organizers very much for inviting me, because I am not a nano guy by any standards. So I was a little bit surprised when I was invited. I thought maybe you got the wrong guy, but hopefully I'll make it up. So now I have to just operate this PC, but hopefully be able to do that. Okay. There we go. All right. So just in case you are a little bit tired after lunch now and you decide to fall asleep, I'll give you a brief abstract of what I'm going to talk about. I'll show you that we can take pieces of sugar, we can stitch them together and make bigger pieces of sugar, we can make structures out of those structures, and if you stay awake, I'll tell you what we do with the structures, but it has to do with nano applications. We talk about carbohydrates. You all know about carbohydrates and nutritional value. You just enjoyed some maybe at lunchtime. We'll not talk about that. Carbohydrates are important because they make up 80% of biomass. Those green things outside called trees, shells of crabs, other things, all carbohydrates. That is important. But also we are all surrounded by carbohydrates because our cells are surrounded by thick coatings of carbohydrates, the so-called glycocalyx surrounds bacteria, parasites, but also mammalian systems. This is the outside view of tuberculosis. But also carbohydrates are important in signaling processes. There's a part in your life that you most likely do not remember when a sperm cell gets together with an egg cell. That involves carbohydrate-protein interactions, and that is not the only such interaction. They also play a role in tumor metastases and others, but just to remind you that signaling is also something that carbohydrates play an important role. Now, when we talk about carbohydrates, then please don't think of sugar pieces, but really think of 4 distinct classes of molecules. And I'd just like to schematically introduce those to you by a textbook view from a biochemistry textbook. That is the outside of your cell, of a mammalian cell. And what you're going to see is a dense forest of carbohydrates, the so-called extracellular matrix. And these long polysaccharides anchored via proteins are so-called glycans. And if you had to go to the hospital, then most likely you did receive shots of heparin. Heparin is an anticoagulant which would never be allowed in the clinic today. It was introduced in 1936 before the FDA of the United States was around. So this is still used today. The market is in excess of $20 billion per year. Then I'd like to remind you that most proteins in mammalian systems are glycoproteins. The market for therapeutic glycoproteins is $85 billion per year. That's a lot of money. And the most famous of those glycoproteins, called erythropoietin, it's a red blood cell stimulant that's made exclusively for these cancer patients shown here. The reason it's used, it makes red blood cells, and that, of course, is abused by some people. So you might say, well, I didn't have to go to the hospital, I don't race my bike, so why the hell do I care about carbohydrates? Well, you should care about carbohydrates because there are oligosaccharides in form of glycolipids anchored in your lipid bilayer that determine, amongst other things, your blood type. So you have these carbohydrates on your surface, you have antibodies against the other blood types, you've been given the wrong blood, it reacts with the carbohydrates, and you die. So these are important for you. What's not yet shown in the textbook is another class of carbohydrates, the so-called Gpi Anchors, glycosyl, phosphatidylinositols, lipids, glycans, and proteins on top. About 0.5% of our proteins in humans are Gpi anchored, and there are 2 laboratories worldwide that can isolate microgram quantities of those Gpi anchors in 6-month processes. So that is a fundamental problem of carbohydrates. To get your hands on pure carbohydrates from biological sources is almost impossible. And that is a shame, because we would like to know more about carbohydrates. As I said, 80% of biomass are carbohydrates, about half in the ocean, half on land. Chitin, the outside of these beetles here, cellulose is important. And for many of you, chemists, material scientists, engineers, you look at this and these are very simple polymers. This is cellulose, beta, 1,4-glucose. The glucose shows up over here again, but now it's not beta, but it's just alpha. So only difference is stereochemistry here. Here you have a crystalline material. Here you have a very soft polymer. So very, very small differences in the structure yield huge differences in the material that comes out from it. If you go to hemicellulose, again, different properties. If you change one of the hydroxyls for an amine that's acylated, then you get chitin. You can get a very, very dense and hard material. So we would like to understand, can we learn the principal rules, and can we then make materials based on carbohydrates? This is now the outside of the cuttlefish, starting from glucosamine, chitin, that forms sheets, that forms coiled coils. Eventually, you get materials. So by now, what you might have realized is the goal of my department, of about 85 people that work with me, is really very, very simple. All we like to do is we like to correlate glycan structure with glycan function. And there are 2 basic technologies that I'm going to briefly describe to you, which we use to try to understand that. One is sequencing of carbohydrates, and the second one is synthesis of carbohydrates. So overall, our goal is to create an automated synthesis platform to make all the carbohydrates I showed you, and do it in such a way that you can do it yourself at home, ideally. Then we like to understand the basic biological processes where these carbohydrates are involved, as well as their structure. And then we like to address medical needs, and we like to create novel materials. For today's lecture, I'll show you some of the basics, and afterwards I'll talk about some unpublished stuff towards the end into that area. But let me take one step back. This is Hargo bin Korana, my chemical grandfather, and he published in 1972 a paper which I think is one of the most important papers ever created because it shows an immense breakthrough for synthetic chemists. It was the first total synthesis of a gene. It was a 77-mer oligonucleotide. He had already received a Nobel Prize a few years earlier for figuring out the genetic code, but he made this, and I calculated this paper goes from page 209 out of page 550 or so. And the total time it took to make this gene was about 35 postdoc years. Okay, so it was not surprising. There was a news and views in Nature that basically said it was a tour de force. Like NASA with its Apollo program, Korana's group has shown it can be done, and both feats, flying to a moon or making a gene, may never be repeated. All of you working in nanotechnology having to do with DNA will know that this is nonsense, because I don't know how long it takes in Israel. In Germany, we can send an email with a sequence, and then we get it back in 3 days by FedEx, and we can go on, okay? So what happened in between? In between happened that Mark Matucci, one of the early PhD students of my PhD advisor, Marv Conovers, figured out that you can make actually DNA on solid support, and introduced what's called the Phosphoramidite Method, the method that's still used today. And that then gives me the reasoning that I argue that the reason we know so much about biomaterials is simply because of synthetic organic chemists. That may be arrogant, but I think it's partially true. Because Marv showed that you can make DNA. And of course, that goes back to an earlier discovery by Bruce Merrifield in the early 60s, where he said, okay, we can make peptides on solid support, because in peptides exactly the same was true. The first total synthesis of insulin was a huge race between huge groups in China, Germany, and in the US. And if you think about it, DNA encodes RNA, RNA then encodes proteins, and proteins encode carbohydrates. And so as a PhD student with Marv, I thought, well, these guys figured out these two things. I should do the same thing in carbohydrates, because they showed that if you do good chemistry, you can do good biology. If you do good biology, you can do good medicine. Marv started 10 companies. One is called Amgen. You may have heard about it. He became filthy rich through it, and you can make a lot of money. And I think with good chemistry, good biology, make a lot of money. I thought it's all attractive, okay? So I figured we'll do the same thing. But for chemists, you will immediately see that there are two fundamental differences between these two biopolymers and carbohydrates. The first difference is this is branched. These are not. Second difference, each glycosidic linkage is a new stereogenic center which can go up or it can go down. So you have to exercise both ratio control as well as stereo control. That's why most people thought this would not be possible. I ended up going to a postdoc to Sam Danishevsky at Sloan Kettering because I wanted to learn how to make carbohydrates. And they had just finished the total synthesis of a hexasaccharide, which is found in women on breast cancer and ovarian cancer, and men on prostate cancer. And 5 people took 2 years to make this molecule. And think about it, if you want to do biology and it takes you 5 people for 2 years, it's almost not doable in most countries because groups like this size don't exist almost anywhere except maybe for parts of Germany and maybe England and the US. So what we did is when I started MIT and we said, well, what we can do is we can automate this process. And we showed that we could do the same thing within 19 hours. So that is the first step forward. I'll just show you a little bit about chemistry. And I should say, if you're interested in the historical part, there's an account which we published a year and a half ago. You can read all that. The idea is very, very simple. You're going to take a solid support resin. You're going to take building blocks. You're going to need some sort of a box, a machine, if you will, that goes through a coupling cycle. You're going to get protected oligosaccharides. You're going to deprotect them, and then you get your materials. So we have now a commercially available set of building blocks that can be put in a commercial available synthesizer. And once you run these two things, then you're going to get more and more complex oligosaccharides, which you can use for all sorts of applications, including possibly in the nanospace. So the idea, again, is relatively straightforward. We use polystyrene resin, so-called Merrifield resin, different linkers we prefer today, the photo-labile linker. We have building blocks. So we have a first sugar on the linker. We couple one building block. We deprotect. We can cap or don't cap. And we can go through this repetitive 2-step cycle. So very similar to what you use from peptide and from DNA chemistry. So I will not go into details and tell you about all the hundreds and hundreds of things you've made. I'll just show you a few highlights. So this one always here really depresses the living hell out of me, because I made this as a postdoc in Danishevsky's lab, 95 to 97, as one of a group of 5 people. My PhD student, Lance Kröck, made this in 2010 in less than a day. Okay, that's nice progress. Then you might say, okay, Seeberger, how long can you go? Well, I'll show you a couple more examples, but Oliviana Carlin, a PhD student, she worked on a different project. And she said, Peter, I think we synthesize pretty cool. I'd like to make something big. I said, well, why do you make a 30-mer? I tell you now what I did not tell her. The largest carbohydrate until that day was a 25-mer made by a Japanese group of 10 postdocs, and she made this one in a week. Now, in all fairness sake, the Japanese made something useful. What we made is probably useless. We just wanted to show it can be done as sort of a proof of principle. So now you might get the idea, the Seeberger lab does anything, everything goes fast and easy, and it all works great, okay? No, just don't be fooled, right? So we worked also on the chondroitin sulfate, heparin sulfates, the glycans with glycans. That's a very, very difficult project. It took us about 15 years from the start. Always a lot of people. Finally, we cracked this. We published this paper, and that was a big breakthrough because we can also now make sulfated glycans, which was very, very difficult because you do not just have to make the linear polymer. You have to afterwards do lots of chemistry. Now, this slide doesn't tell you anything except this is one of the subgroups in my department who works on plant carbohydrate materials. And within that group, one master student, she had never done anything in the laboratory. Her job was to make these oligosaccharides here. She made the building blocks, she assembled them, she purified them, and in 6 months, she got all these structures. Now that would have been easily, in my lab, 2 years earlier, a PhD thesis of 4 years of work. She did this in less than 6 months. I just show you this because that shows this really works now, and you can do it too. You don't need much experience to do this. Then the question is, okay, what's the limit? The record in the lab right now is a 50-mer. We made this, and at that time, to make a 50-mer, we had, without capping, a coupling cycle of about 1 hour 40. With capping, we took about 5 hours. And Naresh Kotari started on December 23rd, the day before Christmas in Germany, and most people go home. And he started the machine because the students wouldn't let him on the machine for 10 days straight. He ran it over New Year's, and after New Year's, we took it off, and we took a spectrum, and it actually worked okay. The yield was only 4%. You might say that's pretty lousy, but it's 4% more than anybody else could get. It by far the largest oligosaccharide, okay? So it works. So what I try to tell you in my introduction here is basically, look, we can make everything. We can make more and more and more and more complex carbohydrates, but the question is going to be afterwards, what can we do with it? And for the last, 10, 12 years now, we thought about what we can do with carbohydrates. I'll show you now some examples. And I think for your conference here, the most interesting part is shown down here, because a long time ago, we started to take carbohydrates, we started to put them on first flat surfaces to make arrays, then we took spherical surfaces, and I can say we have put it on almost every nanoparticle that comes our way through collaborations. We have put it on polymers, we have put it on tissues, anything you want, we have put carbohydrates on. I'm sure there are people in the audience that have good ideas. Please talk to me afterwards. You may be able to collaborate, because I know very little about nano. I know a little bit about carbohydrates. We can also make monoclonal antibodies. I won't talk too much about this. I'll end up today with talking about vaccines in just a few slides to show you what is medically possible. I should say by now, this is not a technology which just we use. It's now being distributed. Just yesterday now, they sold one into Canada. Way means Glyc Universe, a company that some of my students started in Berlin. And we hope at some point that also will be one placed in Israel. I'll be talking to some of your colleagues, but as always, these things take a little bit of time. So I would just like to get to one other point to just highlight it, not to give you all the details, but just to highlight it. So my problem is we can make more and more complex carbohydrates. And we are the Max-Planck, so we are very well funded. But my boss is the president of Max-Planck Society. He doesn't like me because I come and I want a new NMR all the time. Because I make a bigger molecule and I have to completely analyze it as a good synthetic chemist. And I got a 700 now, and now I want a bigger one, right? And there's the end of it. So the question is, can we somehow get an area where people are with DNA and peptides? You do not have to take all the NMR data to analyze. All you do is you take a HPLC, you take a mass spec on micrograms, and boom, you got your sequence. And in the case of carbohydrates, this is more difficult. And typically we have to do mass spectrometry and, of course, NMR, and that takes multiple milligrams of material. And the reason is that there are 3 levels of complexity not associated with DNA and with peptides. The first is composition. That means glucose, this is equatorial, galactose is axial. That is not a problem for us as synthetic chemists, because my students know if they took a bottle of galactose or a bottle of glucose. Okay, that we can solve. Second issue is connectivity. Is it 1,3 or is it 1,4? That for synthetic chemists, again, is not such a big problem because we know where we put our protecting groups. So that is some solvable problem. But the last challenge, and that is configuration, meaning linkage going up, beta, or down, alpha, that is something we cannot always control completely. We like to know, are we pure, or are we not pure, and what do we have in our hands? And so I have asked all the different mass spectrometrists that I know, and believe me, I know a lot of them, and they told me, oh yeah, we can do this, not a problem. We give them stuff, and nothing worked, okay? So a new assistant professor came to our department. He happened to be the first PhD student of my wife's some time ago, and I asked him, he said, Kevin said, oh yeah, we can do this. I said, yeah, sure you can, like all the other guys. He said, no, we're gonna use ion mobility mass spectrometry. And I said, ion mobility, what? And he goes, well, it's sort of like HPLC in the gas phase. And if you think about this, ion mobility basically means you get your materials into the gas phase, you have an electric current, you have a pipe filled with inert gas such as helium, and depending on the shape of your molecule, it's either going to come through faster or it's going to come through slower. And what you get is a so-called collision cross-section. And that is a molecular property that can also be calculated. And that is inherent. So what we did is we made a whole slew of different structures, and I can tell you we cannot, until that point, we could not distinguish glucose versus galactose. We think we may have solved this now, but importantly, we can distinguish alpha versus beta. Okay, but we also want to do it quantitatively, because I don't just say it's alpha or beta, I want to say how much alpha and how much beta, and what we can do is we can go down to about 0.1% impurity. And that is now automated, it's very fast, and it gives us a very, very quick readout. We published this in a good journal, and of course they came back and said, well, trisaccharide, it's not enough, can you go up 25 mers, 30 mers? So we went all to big pieces, we can shoot the big pieces into smaller pieces, and then we can break it down and analyze it. So that helped us tremendously. So what we really wanted to make is we want to make materials. And I showed you that we were limited because our syntheses were too slow. So two people, a PhD student, Andrew Kononov, and Martino Bianco, a postdoc, they looked into the system and they said, okay, can we make this whole process faster? I won't go into gory details now. I can tell you we have shrunk this down to about 1 hour now, and with capping at 1.5, which means in 1 day now we can make a 20-mer. We have 4 synthesizers in our department, and every day on one of these machines, each of the machines, we can make a 20-mer. So that's the rate we are going through. A 20-mer before, I think worldwide, world maybe 3 labs that can make a 20-mer by any standard, and usually that's 4 or 5 people a few years. So that's where we're at now. So the question is now, can we actually make longer structures? Because what I would like to do is, I would like to start to think about, can I build cellulose from first principles? Can I build chitin? Can I start making hybrid materials? Can I mix cellulose with chitin? I have no idea what's going to come from it, but I think it would be cool to try and see and do it. So we have made rows of mannoses, we've made rows of glucoses, but I can tell you when we started to make rows of glucoses, we found very interesting things. They become extremely insoluble. And that's not totally surprising because it's known from cellulose, but it's a crystalline material. However, if you put, for example, a mannose in between every now and then, you start to break the symmetry, and then all of a sudden, you have no longer the solubility issues. So you can actually, through synthesis, have a complete control over physical properties of what you get. And what we want to do, eventually we want to make Lego bricks, which we can then start to mix and match. So what we have now is we have chitins, mannoses, glucoses, and you can again mix in different things. And we then work on block couplings, where we have, let's say, a 6-mer, 12-mer, 18-mer, 30-mer, if you will, and then we can start putting this together. So, of course, you could do this also on disaccharides, but in that case, you make a polymer of linker and not a polymer of polysaccharides. So that is something that we are in the process of making right now. We have finished many of these structures, and they behave, in many cases, very nicely. In many cases, they behave very badly, okay? They fall out of solution, they become awfully crystalline and very difficult to deal with. So we're learning a lot about basic principles. We think at one point we may be able to even do things like carbohydrate origami. We might be able to fold carbohydrates, okay? The problem right now is we don't know how they fold and why they fold, so we've done a lot of theoretical studies also trying to understand the structure of carbohydrates. Okay, then you can start to think about carbohydrate as multivalent scaffolds. The carbohydrates are typically on the surface, they interact in multivalent fashion with other cells, and people have done a lot of work in these areas, and we reviewed this last year in ChemRef. But basically, you can take organic macrocycles, such as cyclodextrins. You can use dendrimers, metal dendrimers. This was in our lab, started by Raghu Kikari, who got his PhD with Avi Shancer here at Weizmann. We've put it on graphene, we put it on quantum dots, on nanoparticles, on appended polymers, and you can make copolymers. And they're the only ones. Many people are working on this. I'd just like to show you a couple of applications. So for us, what we have done is we made cyclodextrin hexameric complexes. Oh, it's a very poor solution here. We have put it on graphene. We put it on nanoparticles and on gold nanoclusters. We have used it for brain stroke MRI, and we've looked at cancer cells. So this is work by Dan Grünstein, PhD student, who finished a few years ago in my laboratory. And the idea was basically to create sensors. This is done in collaboration with Israel Rubinstein at Weizmann, where we had ruthenium in the base, have ligands around that, have an adamantine out here, cyclodextrin. On the cyclodextrin, you have 7 different carbohydrates you can place, and then you have sort of like octopus type situation here, and that can interact with different E. coli, and you can sense things quite nicely. It's a modular system because you can change the bases, you can change the carbohydrates out here, and you can change the valency. The same sort of principle was done with Rainer Haag at the Free University of Berlin, where we used modified graphene surfaces, so sheets now of graphene modified with adamantyl groups out here. The idea is the same again, cyclodextrin with carbohydrates on the surface. The idea is to have this multivalent display of carbohydrates. You have E. coli. E. coli bind specifically to mannose. The idea is, that basically you have the E. coli binding to the mannoses, and then you could detect or kill it. We have 2 strains of E. coli. Orndorff or N208 is a mutant that has no pili, it has no binding proteins to carbohydrates, and ORN178 is a normal infective strain of E. coli, and what you can see is this stuff doesn't bind very well, and this stuff is engulfed by the graphene sheets. So if you take a closer look at that, you can actually see that the bacteria are sort of wrapped like a present in graphene in those cases. These are more detailed pictures now on the inside. You can basically see the cells that are being wrapped by these graphene sheets. That's all good and fine. It's sort of like a toy at that stage, but you can then take a look at the confocal laser scanning microscopy. And what you will see in this case is that if you do 178, you can actually see a nice wrapping. If you take the bacteria without the mannose binding properties, then you no longer see these kinds of interactions. The idea was then, could we use that to actually clean surfaces and kill bacteria? So what we see here on the left side is just E. coli in here. We measure internal temperature here. We see a temperature, just room temperature basically, probably Germany summer or something. And then you can do the same thing with the graphene, including the mannose on cyclodextrin. And what you see here now is this heats up drastically, because basically what you're doing is you're harvesting the energy on the bacteria, and you basically burn off bacteria. So the Free University filed some patents around this. They think they can use this for cleaning surfaces. I think it's a little bit difficult, but it's one potential application for these graphene surfaces. After IR laser irradiation, what you see is E. coli without irradiation, and this is E. coli with irradiation over here, so you have a clean plate afterwards. In collaboration with a former postdoc of mine in China, at Shanghai University, the idea was now to think about delivering doxorubicin, a drug, as a just example, and the goal would be again to take cyclodextrin, put carbohydrates on the surface. Carbohydrates will be placed on the outside. Doxorubicin is in the middle, 7 mannoses on the outside, and then the goal is to basically engage receptors on the surface of cancer cells, have receptor-mediated endocytosis, and then have an escape by pH from the endosomes and get a doxorubicin release that then in the nucleus does its job. So that was a goal. When you make the particles, this is the beta-cyclodextrin nanoparticles before drug loading. This is after drug loading, so you see a difference also in the hydrodynamic size from 112 nanometers to 199 nanometers shown over here. What's important is the stability and release based on pH. This is before drug loading, after drug loading, but then important for pH at pH 7.4, quite stable over many hours, but as you go to lower pHs in the acidic medium, but is prevalent in endosomes, you then see a breakage and release of a drug in this case. If you now, it doesn't come out very well at all here, if you go and take a look at this by having the molecules plus the cancer cells plus mannose, you can maybe see a little bit of a red halo around here on the screen. You would see this very strongly. If you put in mannose for competition, so there's no more interaction, this can be downregulated. If you use it in normal cells, then you don't see that either. So it's very specific for the cancer cells. This was tested in vivo in mice in this case, so it can either take saline or different combinations, or what you see is you can actually use the doxorubicin with the MAN7 cyclodextrins, and if you just take a visual inspection and you look at the tumor size, if you give saline, nothing changes. If you take just the CD mannose, nothing changes. Doxorubicin, not too much happens. With glucose, nothing really happens. And if you take the one loaded with mannose and doxorubicin, then you get a specific effect. So you can actually deliver doxorubicin in vivo into the animals, and you can see an effect on the tumor. So that is work again done in China, and the Chinese have now applied for Chinese patents and are moving this forward based on our work. In my own laboratory, Silvia Varela Aramburu, a Spanish PhD student, by accident came across a method to make gold nanoparticles. This is relatively simple. You start from a gold salt with a glucose thiol in water at room temperature. You get thiol substitution on gold, and you have carboxylic acid remaining out here. It's a non-toxic and cell-permeable molecule of 2 nanometers. It's a one-part synthesis. It's very simple. Anybody can do it on large scale. These are basically highly monodispersed particles, quite small actually particles. They are stable, and we can functionalize the surface as shown by fluorine NMR and agglutination assays. They are non-toxic and they are cell permeable. So what we are now trying to use is to use those nanoclusters that we can make from gold, put a dye on there for imaging, put a drug on there for nanocarriers, and put a targeting moiety on there. And what we are targeting are our favorite targets. We have worked for many, many years on Plasmodium falciparum, the causative agent of malaria. We have worked a lot on Toxoplasma gondii, and we are working quite extensively on tumor cells. And for tumor cells, there is one effect that is relatively poorly understood, and it's not completely agreed in the community. You heard of protein-protein interactions. You know DNA interacts with each other, and some people believe that carbohydrates also interact with other carbohydrates. And that is something that not everybody agrees. And I was agnostic. I didn't know. I said, okay, let's test this. And this is a molecule of tricyclic called GM3. It's a ganglioside. It's present on melanoma cells. This is G3, another ganglioside. For most of you, looks almost the same. There are slight differences, okay? But it was believed that these things interact with each other. The problem was that biologists usually use entire cells. They try to bring these together, and then you get proteins on top, you got all sorts of carbohydrates. It's a little bit difficult to do. So Jiren Lai, the postdoc, now professor in Taiwan, she wanted to look into this, so she made those different structures, and we wanted to see if this is really true, because people thought that this was the initial step of adhesive process in tumor metastases. So what she did is she basically took silica nanoparticles, which we got from the laboratory of Luisa De Cola from Strasbourg. We placed here, through coupling, the sugar on the surface. We did the theta potential, dynamic light scattering, and we placed different glucose, lactose, Gg3 on there. So we can make a GM3 polymer slide. We take our silicon nanoparticle and we can do an SPR study. If it is glucose, there's no interaction. If it's lactose, there's no interaction. But if it's Gg3, there's indeed interaction. It's not great, but there's clearly a specific interaction that helps. And that was, to our knowledge, the first time somebody could actually show, using not a biological sample, but a nanoparticle, that indeed there are carbohydrate-carbohydrate interactions that can also be measured. So that, I think, was important from that perspective. This can also be looked at in cells. I apologize, this really comes out quite poorly, but if you just allow me, if you will say you take your Gg3 and dye manipulate silicon nanoparticles, you can actually stain cells. So it's not something that happens only on an SPR slide. This is something that also works in biological samples. Now, most of the things we do in my department are a little bit applied, so we wanted to see, can we actually enable people to use this in the medical field? This is work with a MD at the Charité Hospital in Berlin, a neurosurgeon, and what we wanted to do is we wanted to see if we can actually image the early endothelial inflammation that follows a stroke. So after a stroke, there's typically inflammation in the brain, and the idea was that possibly we can actually image this whole thing by putting a ligand on there, a ligand that binds to E-selectin. So E-selectin is a part of the inflammatory response. E-selectin is known to have carbohydrate ligands, and what we wanted to have is basically image that through these dyes. For that, we need a rat model. We have to induce a stroke in the rat, and we're going to follow in the rat, in the brain, and trying to image this. So first of all, these silica superparamagnetic glycan nanoparticles were looked at. E-selectin binds to them. You can again look at the size of molecules. That was done in this case. And then you can look at the MRI. Looking at the MRI, you basically take a rat at different time frames. You're going to induce the stroke. And what happens is with different nanoparticles, this is now just hydroxylate nanoparticles, you don't see very much. This is when another kind of nanoparticle, again not so strong, but the carboxylate nanoparticles, you can actually start to see this also in Western blot. So these types of things are actually binding. So by now I realize most people have sort of checked out because they said, okay, Seeberger, all he's going to do is just take one spherical thing after another, and he's going to just stick carbohydrates on. That is exactly true. And that's why I stopped this for the most part in my lab. Because we show to ourselves, you give us a spherical surface, a nanoparticle you made of any kind, we can put carbohydrates on, and we're going to see something. You're going to publish a paper in a reasonable journal, but I'm sorry, I'm not that interested, okay? So I dissolved a large part of my nano group because of that, because I said, okay, if you guys don't think of anything new and new materials, then I don't want it. And so what I can offer is, if you have systems where there's something interesting where carbohydrate could work on, we can provide carbohydrates, we can also functionalize things, we can work with all the tools in hand in our institute to do it, but there has to be a cool application and a great paper afterwards. Otherwise, we don't do it, because we have shown it so many times, okay? And the same holds true for the next two slides I show you. My lab was the first lab to make glycan arrays. 15 years ago, shortly after us, Chiwei Wong did it. It's now a staple, everybody's doing it, okay? And the idea is very simple. You take a flat surface, you take a carbohydrate, you stick it on there, it's going to bind, and then you're going to see other stuff that binds. It's been done for DNA, it's been done for peptides, and by now you may have realized that I'm not creative, okay? I look not only what people do in DNA, I look what people do in peptides. We do the same thing in carbohydrates, and we get to publish papers. It's so simple. But the question is, what can we do with it? What we have done now over the last few years is we have created, through automated synthesis, a very large number of carbohydrates. These carbohydrates are in a database that we can pull from. We can print custom designed arrays, and we can use these arrays to look at biological samples, to look at binding patterns. So we have done things like purified proteins. We have taken every biological sample except for sperm and have tested it, okay? You can do it. You get very, very interesting results from a biological standpoint. You can take whole cells and viruses, and we've published, I think, 80 papers or something on carbohydrate arrays. I would just like to highlight the power on one single example. And maybe that's time to wake up for a second. Because for 50% of you, this is an issue. Because 50% of you living in Israel, me living in Germany, most likely carry toxoplasmosis. Toxoplasma gondii is a parasite, protozoan parasite. It is very commonly distributed. It's more in countries where people have cats at home, because cats carry it. And it's more carried in countries like France, where we eat beef tartare. If you eat raw meats, chance of contracting toxoplasmosis is more than in other places. Don't worry about it. Not going to do anything to you, unless you are a pregnant woman and having a first infection during pregnancy. That can lead to severe damage to a fetus. This is not a joking matter, this is serious, okay? The problem is that in Germany, for example, testing for toxoplasmosis during pregnancy is not covered by the health services, by our national health insurance. It's an individual health thing that if you want to have it done, you have to pay for it. It costs about 250 euros for somebody to do this. It takes a few days, and this is not a lot of fun. I realized it because my wife, when she was pregnant, her second child, she was scratched very badly by our neighbor's cat, and she was worried. She had to go to the hospital, be tested, and 3 days later you find out, okay, everything was great, no problems, but it's something you would like to have it faster. And at the same time, we started to think about this, and I said, okay, couldn't we think of something to make this faster? And the reason we got interested is because on the surface of these parasites, there are these things called Gpi Anchors I mentioned at the beginning, lipids, glycans, and in toxo, there's a specific side chain out here. So I've worked on this for 15 years on these carbohydrates, so we made the structure. Don't worry about the details here, it's unimportant. Basically, the idea was, is it possible that humans make an antibody response against this particular sugar structure? So what we did is we made arrays containing that structure, but also closely related relatives. And long story short, we can tell if it is IgM or IgG antibodies, so we can tell if it's a fresh infection or if it's a infection that happened previously. If it's an infection previously and the woman had been already infected, not a worry because she's going to be fine, okay? Second infection doesn't matter. If it's a fresh infection, we have to worry. And so what we can do is we can do this on a chip that's about this size. We need 10 microliters of blood and about 5 minutes. And we can read it out with a cell phone. So together with the company we work with, we can do this now. We can make a finger prick. It pulls a little blood on the chip, and it can be read out by a cell phone, sent to a database, and about 10 minutes later, you know what happened. This test kit costs about €1 to €2, and it would be very, very cheap to make this. We patent it all. We got European money to actually make a company out of it. We haven't done it yet because it is unclear whether there's a business model for this to be done, because the doctor has to make money. And right now, the doctor, by prescribing it, makes a lot of money because in the food chain, a lot of people make money. But if you do this, you could actually help people very great. But most likely, the doctors would make less money than before, and that's why it's unlikely that somebody actually use it. So scientifically great, unfortunately, economically not so viable. With that, I'm almost at the end, and I'll come to something that hopefully nobody in the room yet probably, but your kids or grandkids hopefully have received carbohydrate-based vaccines. In Israel, like in Europe, like in the United States, the first carbohydrate-based vaccines came on the market in 1992 against Haemophilus influenza type B. Most of you probably never heard of HIP, and it's good that way, because if your children would have had this, the chance of dying from severe disease in those cases was very, very high. This is a very serious bacterium. You don't want it. And that vaccine has saved millions of lives of children. The second vaccine that came around was the early 2000s against meningitis, and the big blockbuster came in about 2006, 2008, when Pfizer put Prevnar on the market. Prevnar in 2015 made $6.4 billion in revenue. And I mention this to you because it's very, very important for our field. Because I worked on vaccines for the past 20 years. And people used to say, Seeberger, come on, you work in vaccines. Nobody makes any money with vaccines. You're never going to get any of this stuff on the market because nobody makes money. If people don't make money, they don't fund it. With this happening, everything changed, okay? It changed because millions of people have survived pneumonia, meningitis, and other things, but also people make lots of money. Now, the way these vaccines work are very, very simple. The whole basis for vaccine is that somehow you have to educate your immune system to recognize something foreign, and when it's recognized, it has to be killed. And the way it was done a long time ago is you take the whole bacterium, you kill it, and then you inject it. Or you could take a piece of it. And that's what's done in these carbohydrate vaccines. I told you in the beginning, many of bacteria are surrounded by carbohydrates. So what I do is I grow bacterium, I harvest the carbohydrate, I purify it. In the case of Prevnar, that took Pfizer 20 years to figure it out. 20 years and 13 strains are in there. Now, the problem is, from an economic standpoint, that this is only protected by process patents. It's very easy to get around process patents in China and India. It's very difficult to get around composition of matter patents, but that's a side effect. The second thing is that if I want this to be used in small children and old people, main constituency that need vaccines, then I have to get an immune response. And small children cannot make an immune response against carbohydrates. And that's why all these things are connected to a carrier protein. And that makes these vaccines temperature sensitive. And you have to maintain a cold chain. In Germany, that's not a problem. It's always cold anyway. But most other countries worldwide, that is a huge issue and costs about 50% of the cost of the vaccine is the cold chain. And finally, you have to have an adjuvant, and this is only an injectable. And that's a big problem in many developing countries because they reuse the needles. So what we said is, we would like to completely and radically change the way vaccines are made. We want these not to be biological. We want these to be completely pharmaceutical compounds. Synthetic carbohydrate, synthetic glycolipid, no adjuvant, and we like to be able to just use it and smear it in the nose and get the same effect if I make an injection. I will not talk about the fact of no adjuvant. This is published in Nature Chemical Biology 2014. I will not talk about nasal delivery, but I will just give you one example of how we can think about such vaccines. I should say that in our department, we basically can do everything from the synthesis all the way over to experiments in animals. We have our own facilities up to a safety level 2. We can do infection experiments. We work on things that are important, such as leishmania and malaria. We work also on stuff that's totally unimportant, like plague. But let me tell you a secret. It's really easy to get funding for biowarfare. It's more difficult for these other things. And so what we do is, don't tell anybody, okay? We take the money we get from here and we use it here, okay? Don't tell anybody. So we also work on things like hospital-acquired infections, C. diff, important, and many other things. Some of them are not disclosed yet. I won't go into details. I just would like to give you an appreciation of what the process looks like. So basically we look for a disease that we think is important because there's a medical need. We're going to try to get some samples, typically patient blood or patient stool in the case of C. diff. We're going to make a whole bunch of carbohydrates, a whole library. We place them on a chip. We're going to see if these patients made antibodies and there's a correlation between antibodies and outcome of disease. From that, we select a candidate. The candidate gets connected to carrier proteins or glycolipids. That gets introduced to the animals. We check the immune response by ELISAs, SPR, and glycan arrays. We do functional assays such as OPA, so killing assays. And finally, we're going to challenge the animals. So we have done this many, many times in many different diseases, but I like to just pinpoint Streptococcus pneumonia. And I didn't want to work on this because Prevnar exists, a huge drug, huge success. So what can I do? Well, what I realized is that all of us, as you sit here today, and me, unless you took antibiotics this morning, you carry Streptococcus pneumonia. Don't worry about it. Not going to kill you today, maybe tomorrow, we don't know. No, don't worry about it. But Streptococcus pneumonia kills more children in Africa than malaria and HIV combined. Most of us don't know that. It kills still in all the industrialized countries, Israel, Germany, United States, it kills millions and millions of people. Even though there's Prevnar. Because the problem is that there are at least 96 different strains of Streptococcus pneumonia. And in Prevnar there are 13 strains. And there's a study from Israel. Israel is one of the most broadest vaccine coverages worldwide. And as people are vaccinated with Prevnar, those 13 serotypes are gone for the most part. But what happens is your nasopharynx, where the Streptococcus sits, is cleared of those 13 strains. If the other 83 strains, hey, fantastic, lots of space here. Let's move in and let's make people sick. So what you get is you get serotype replacement. And that's a constant race. So we always have to increase the coverage of different serotypes. This is the prevalence of serotypes, Prevnar 7, Prevnar 10, Prevnar 13. And the problem is that even in Prevnar, some serotypes don't work very well. You can see this, you go to hospitals, you see that certain people get sick. If you're a cynic like me, you might say, why is serotype 2 not included? Because serotype 2 mainly kills small children in Southeast Asia, where we don't have money to pay for vaccine. I don't think that's the reason, I think there are different reasons. Serotype 3 doesn't work very well in the new vaccine. And so about 5 years ago, I said, okay, I like to make a streptococcus vaccine with 25 serotypes. It's easy for me to say that, but it's a different thing to do this. So we had 14 postdocs put on a project. We said, okay, now we're going to try to go after this. And I apologize, it's a little bit weird drawing here. But basically, any one of those molecules is basically a series of total syntheses. We have reduced them to practice in many, many cases. And in some cases, we've gone very far. If you look at Science Translational Medicine the day after tomorrow, we have a vaccine coming out for Streptococcus pneumonia serotype 8. That's the main killer right now in the United States and Europe. I don't know about Israel, but it's a huge problem. We have something very advanced right now, which is actually going towards human clinical trials, but we've also done this in other areas. I'd like to just show you SP3. SP3 is in current vaccines, which many of us may have gotten, but it's not working very well because there are issues of immunogenicity. So what we did is we selected a tetrasaccharide. We then vaccinate animals 3 times, and then we, and in this case, with a pulmonologist at Charity Hospital, the pulmonologists basically take a syringe with bacteria, they stick it in the nasopharynx of the animal, and we watch the animal. Those animals that are vaccinated clear bacteria within 2 days, and they are perfectly fine, and they get a complete restoration of lung function. This was published last year. Those animals that do not have this vaccine basically die miserably, much like we would if we would get infected. So now I told you that we have really great vaccines for mice. And you, maybe like me, just don't care. Because I don't want to make vaccines for mice, I want to make vaccines for humans. So the question is, how are we going to do that? Max Planck is really great, get lots of funding, wonderful freedom to do research, one of the best places I've ever been, better than MIT, better than ETH. So how can we move forward? The only way we can move forward is either by licensing it out or creating our own company. We opted for the second option. Took us a little of effort, but finally 2 years ago we got 40 million euros from Actelion, a large Swiss-based company, to start Vaxilon. They licensed a portfolio of about 25 patents from us. They put 20 people in place, and they have now actually clearance by German authorities to move this into clinical trials. Just last year they received the Top Start of the Year award in Germany. They are now off to races, and within the next 8 months, this is going to be tested in humans for the first time, and they have a large series of antigens moving forward. I was given a sign 2 more minutes, so I figured we should take the last slide. What I wanted to tell you today is that automatic glycan assembly accelerates the process of making carbohydrates greatly. It's today commercial. You can buy the synthesizer, you can buy the building blocks, you can also buy the oligos from a company I mentioned. We can make ever and ever more complex carbohydrates. We cannot yet make everything, but we can make lots of stuff. We begin now to do material science on carbohydrate bases, just starting. We work on the glycoarray platform, another spin-off came out of that. We work a lot on monoclonal antibodies and also on nanobodies, I should say, in the Negev. One of my postdocs, Oren Moskowitz from Weizmann, we have several alpacas that have our carbohydrates in them, which we bleed and actually look for nanobodies. And I showed you a little bit about our vaccine programs. As always, the most important slide comes at the end, because I direct the department. I try to put in my time in research as much as I can, but the people actually do it every day, 24/7. The people shown here. The people who work directly with me is about 20, 25 people. The whole department is about 85, ranging from engineers and chemists over to medical doctors. They represent, at this moment, I think 18 different countries. I'd like to keep it a little bit diverse with us. This should be much, much bigger. That's the Max Planck logo, and Max Planck pays a lot of our money. About half the money comes from Max Planck. The other comes from many other funding sources, ERC advanced grants, other things. I really like to thank them. Fantastic group of people. Many have gone on to academia, some also back in Israel successfully. These are electronic means by which you can contact me, but I think if there's still time, happy to answer your questions. Thank you very much. Okay, so if you think about this, this goes to stepwise. I'll repeat the question. Sorry. So the question was, is the yield typically only 4%? What I said is the yield is 104-step synthesis, 102-step synthesis, 50 mR. The coupling yield today is greater than 99%. Otherwise, you cannot get to these kinds of yields. So if you go to something that you might use regularly, 5 mers, 8 mers, then if you do it on a 25 micromole scale, which is a typical scale we do, 12.5 or 25, you get about 50 milligrams of material. For us in the vaccine space to do everything up to animal experiments, we say internally we need 18 milligrams of material. So one synthesizer run is enough of that. So the typical yield is greater than 99%. And so it depends again on the length of what you do. It's not like DNA synthesis where you get 99.999%, but you're getting close to peptide synthesis in most cases. Okay?
Fantastic talk. You showed some carbohydrates targeting MBA and beta-3-1 cells. What were they targeting there? Which molecule, which receptor?
I think this is, there's a mannose binding protein up there, which is, I think, there are langerins up there that are bound, but I think there are multiple molecules that bind actually mannose. And to me, that is the main issue. The main issue is that it's for very few of those lectins, specificity is known. So we had two groups in my department, Bernd Leppeny's immunologist who worked with me, is now professor in Hanover. He looked at a lot of carbohydrate binding lectins. And what we like to tease out are fine specificities, because if you're going with mannose, I think this is a proof of principle, okay? We always use mannose because it's easy. But what you really want is, I think you want something bigger, you want something more specific, because otherwise you're going to get whole subsets of stuff that bind. And I think that is really something that's plagued the whole field. Bernd Lepeny is now Christoph Rademacher in my department. He looks a lot at langerin, by the way. They look at this. But the problem is the moment people find a ligand, then they usually go very deep into biology and they never come out again. So because it's interesting what happens afterwards, right? But what we really need is we need a better understanding of all the different specificities, because then you can actually start targeting better. But I would say 98% of everything that's published is on mannose, because what people can make, right? And the other 2% is a smattering of other small stuff. What we really want is a much broader basis on what's actually bound there.
Another question. Yes.
You made a lot of emphasis to medicine, but is there also some big hope to get to a new important material in the synthetic process?
So the question is there a way to get a new important material to new important materials?
In terms of structure, materials.
So we are getting to this right now, right? What I try to show is this Lego type thing that I showed you, where we start putting things together. I'm extremely excited about this. So we are just right now writing up the first paper in this area. It took us only 4 years to get to this point. I think we are now at a point where we can make, for us, big quantities, let's say 100 milligrams of material. I think from an application standpoint, by synthesis to make materials will be, in the beginning, prohibitively expensive. Because think about peptides, right? Peptides are 50 years ahead of us. It's still difficult. But I think what we're going to get is we're going to get a better understanding as to how single modifications change materials' properties. And then maybe we could go back to cellulose that's extracted, to chitin that's extracted, and then make those changes, or use enzymes to do that. Because I think by total synthesis, to make a material at a cost that's cost sensitive, I don't see that, okay? For vaccines, it's perfect because we need microgram quantities, so the cost of goods is irrelevant. I think scientifically, we're at a very exciting stage right now because we can make this. And right now, one of my former coworkers, Matan Hurevich, is at Hebrew University, and I think he's getting very close now to get a...
Even if it's in your joint or in your heart, it's still worthwhile. I mean, not to build huge structure of it, but
yeah, the problem is what's in your joints in your heart is still big, right? Because the gags that are in your joints, those are 200, 300 sugars long. So I think one of the ways one might, of course, people have looked into this, right? Today, it's all limited by the fact we don't know what length we need. So typically people like monosaccharides or disaccharides, they make a polymer and they stick something that looks like a gag on it and they say, oh, it's a joint replacement. Look at it from a synthetic standpoint and say, okay, maybe that's not it. Maybe you need to come a little bit closer to that. Maybe make larger pieces, start fusing with polymer chemistry, with surfaces, and then think of hybrids. So that's why I said hybrid materials. I think taking lessons learned from polymers, combining that with carbohydrates, that's where we like to go. But I'd like to get across to this crowd, if you remember nothing else, Next time you see something, think that maybe a carbohydrate could be an option, because so far people have not touched it because they thought it's prohibitive. And I think what I'm trying to say is there's huge opportunity, and it can be tackled now, and we should not look at what's going to be application afterwards, because that's not going to be there immediately. I think it's going to maybe take a little longer to get there, but the applications are huge.
One last question.
Yeah, so you mentioned, thanks, great talk, by the way. So you mentioned a little bit about origami, or trying to do origami structure with carbohydrates, and the point is that with DNA, right, was the issue of coding, that you can play with the coding and put things together. Is there something equivalent to that?
I don't know. Okay, I mean, origami I use and it worked. Okay, I used it to provoke people. What we find is interesting. What we find today is that if you take mannoses, which we always used as a starting point for us, just because we had mannose around a long time ago, so we always use it as sort of like poly-T in DNA synthesis. We just made mannose oligomers. Mannose oligomers apparently are rigid rods, which is great because they don't form structures. You go to glucoses afterwards, they form something that looks probably like a helix in many cases. Chitin, similar, okay? So the first thing you're doing right now is, and we're learning the hard way, because we make these things, they work great in the protected form, we deprotect them, and boom, the stuff is gone. We thought, where is it, right? Turns out, actually, it forms crystals and all sorts of other weird stuff. So what we are learning now is, we are learning that some of these things form structures. We work with theoreticians in Spain and with NMR spectroscopists, and we are trying to understand why do they form structures and what do the structures look like. What I think right now is that we have rigid rods, we have coils, basically random coils in some cases, and we have things like sheets and helices. I personally don't think that we have something that is really sort of encoding what we have in DNA. I don't think so, a different purpose. But I think we can, in part, get things that behave more like that. Maybe like, say, like peptides, I would say. I think DNA is something that's a different job, right? It's transfer of information from generation to the next. That's a different job from carbohydrates. But I think we'll be able to make things that behave more like peptides in those cases.
Thanks the distinguished speaker, the keynote speaker again. Okay, so